Hey all, welcome to ShareTreck. This is your host Raj. Today I'm going to talk to you about Allogene and Arbor Bio Technologies. They have a new partnership that may provide Allogene with powerful tools to craft their CAR T therapies for autoimmune diseases. Let's get started. Welcome back, friends. Privately held startup Arbor Bio Technologies is based in Cambridge, Massachusetts, and it has developed cutting edge CRISPR gene editing technology and has entered into a non exclusive global gene editing license agreement with Allogene Therapeutics. I would categorize Arbor Bio Technologies as the next generation gene editing company based on what I have seen on their website. It was founded by Fang Zheng and David Walt. The company combines CRISPR technology with advanced AI driven discovery, high throughput screening, and robust protein engineering. Speaking of Feng, did you know that Feng Zhang has founded at least 4 companies? These include Sherlock Biosciences, Arbor Technologies, Editas Medicine, and Beam Therapeutics. Arbor have developed a proprietary discovery engine that has helped formulate a diverse portfolio of novel genomic editors, allowing for innovative gene editing applications beyond simple knockdowns. A quick review of their website indicates that Arbor is working towards a future where gene editing includes reverse transcriptases, nuclease excisions, and large insertions, offering potential treatments for a wide spectrum of genetically defined diseases. And the recently announced collaboration between Allogene and Arbor revolves around integrating Arbor's proprietary CRISPR gene editing technology into Allogene's next generation Allo CAR T platform. Let me elaborate this further. First, let me refresh your memory. PAM stands for Protospacer Adjacent Motif. This becomes a very important aspect for any CRISPR editing. And it's a short DNA sequence that CRISPR associated proteins recognize. Arbor Biotechnologies claim to have a suite of enzymes or nucleases that can cut DNA with diverse PAM sequences, allowing them to target more than 93% of human genome. This is significant because different regions of the genome have different PAM sequences, and having diversity allows for a broader range of gene editing possibilities, opening up a whole suite of tools that Allogene can use to differentiate themselves from the competition. The Arbor Toolbox is a collection of gene editing tools that Arbor possesses. These tools are enzymes and proteins that can modify DNA. The toolbox enables a variety of functions ranging from simple knockdowns, that is, reducing the expression of a gene, to more sophisticated and precise ways of editing the genome, which could include inserting, deleting, or modifying specific DNA sequences with high precision. And remember, they can now target 93% of the human genome thanks to the various nucleases they have. So Arbor Technologies have chosen to concentrate their efforts on 4 primary platforms for editing DNA. At this point, I would like to take you to the website of Arbor to show you the discovery page. So here we are in the next generation editing page for Arbor Bio. And what we have here is the 4 platforms that Arbor is concentrating on. The first one is sourcing from nature proteins and indexing them and curating them. They have around 3 billion plus proteins that they have indexed to date, which means they know the properties of these proteins. They also have artificial intelligence and machine language optimization for high throughput screening to identify and optimize editing technologies. And they are able to generate new discoveries with novel properties. And they have discovered 9 proprietary gene editors with 6 times more nuclease families than are published in literature. So that's a big differentiator for Arbor. And then they are able to engineer and significantly improve editing efficiency by taking the wild type nucleases and making subtle modifications to them and create nucleases that are way more efficient, 30 times more efficient than the wild type for editing. So this is what they are focusing on. These are the 4 platforms that they are focusing on. As we saw, each platform has unique properties that help distinguish Arbor from others, allowing for a differentiated approach. This means that Arbor can apply specific tools and techniques tailored to the characteristics of each platform. This partnership aims to help Allogene advance the development of highly effective and widely accessible CAR T therapies for the treatment of autoimmune diseases. Allogene recognizes the growing interest in CAR-T as a therapeutic option for autoimmune diseases and emphasizes the need of advanced technology for differentiation in this rapidly expanding field. Zachary Roberts, who is the Executive Vice President of Research and Development, as well as the Chief Medical Officer of Allogene, wants to use the Arbor platform partnership to develop a differentiated allo-CAR-T approach for autoimmune disease treatment. Allogene anticipates initiating phase 1 clinical trials for its first autoimmune disease, allo-CAR-T investigational product, somewhere in the early 2025s. In conclusion, I will say that the partnership between Allogene Therapeutics and Arbor Bio Technologies signifies a strategic alliance between industry leaders in gene editing and CAR-T therapies, and this collaboration aims to bring forth groundbreaking advancement in the treatment of autoimmune diseases, leveraging Arbor's advanced gene editing technologies and Allogene's expertise in CAR-T research and development. Though this is a non-exclusive partnership, this ensures that Allogene has access to cutting-edge technology to develop CAR-T for autoimmune diseases. This means we will have to watch Allogene closely going forward, because it will be interesting to know which therapy enters phase 1 clinical trials in early 2025. And if you recollect, I had mentioned in an earlier video that Arbor has tied up with Ginkgo to use Ginkgo's high-throughput mammalian foundry to optimize novel gene editors with improved functional characteristics by screening in mammalian cells. Please let me know what you think of this new partnership, and especially I would like to hear from those of you who hold Ginkgo Bioworks or you are interested in Ginkgo Bioworks. I would like to hear from you when you look at the discovery engine of Arbor out here, and like to hear from you what you think are the prospects for Ginkgo. So that's all for now. I'll be back with the next video very soon. See you later. Bye.